So, welcome to uh, the talk on plant probiotics. We are talking about plant probiotics in our previous class, and I hope you are progressing well on writing the review because it is very critical for you to learn as well as to make a productive work from your learning through this course. Let me show you some of the slides. Probiotic bacteria, when we uh, applied as a seed priming in our previous talk, I told you that they colonize properly on the root, plant root, and dominate in the rhizosphere. And this is an evidence of how probiotic bacteria colonize in the rhizosphere of rice. We inoculated rice root by dipping into the suspension of Acinetobacter, one very important uh, probiotic bacteria. And we checked different parts of the root and the root hair zone, and we found that Acinetobacter nicely colonized and dominate in the rhizosphere of rice. And this is why it impacts on the growth and development of the rice plant. We tested some of the probiotic bacteria in the growth on growth and yield of rice, as well as root colonization in our uh, BSMRU experimental field. And we found that Serratia marcescens, Micrococcus endophyticus, with half doses of fertilizer uh, gave almost similar or even better yield than the full recommended doses of NPK fertilizer. And this research is very convincing and clearly indicate that by using probiotic bacteria, uh, 50% of NPK fertilizers can be reduced with high yield increase. And it is an amazing experiment, and we conducted in the field of BSMRU. And if it really happens in the practical field, means in the farmer's field, then it would be a big impact on our, agriculture because it will reduce the production cost as well as reduce the use of synthetic chemical fertilizer, which are a bit hazardous for the environment as well as expensive. So we can save lots of money. In Bangladesh, government offers huge subsidy to the fertilizer. Uh, and it is a national burden, and especially the nitrogenous fertilizer, mostly prepared by the utilization of natural gas. Uh, and natural gas reserve methane gas is limited in our country, and P and K fertilizers are almost all imported from other countries. This is why NPK fertilizer supplementation by using probiotic bacteria in rice would be hugely impactful. We also tested in the field condition. This is not the figure actually. Uh, uh, in other image previously I showed. Uh, in Ginaido, and we found similar results. And similar results are found in wheat also. In case of wheat, dry matter production by using half doses of fertilizer also significantly increased than the full doses of fertilizer. That means probiotic bacteria are equally active on other crops. Similarly, we used uh, probiotic bacteria, lots of probiotic bacteria we tested on strawberry, because strawberry, we directly eat the fruit. Synthetic chemicals especially the pesticide application, is hazardous. We used the probiotic bacteria as biocontrol agent as well as plant growth promoting agent, and we found that here you can see in case of control, uh, the yield of average yield per plant was 193 gram, but in case of probiotic bacteria, it was much better. And when we tested in the randomized block design in a large field condition, we found that plant probiotic bacteria, especially belonging to the genus Bacillus and Parabarcolderia, improve the growth, yield, and content of antioxidants in the strawberry, because raw fruit, fresh fruits of strawberry uh, offers us huge antioxidants and vitamins and minerals. Those antioxidants, especially phenolic compounds, vitamins, and minerals, they are very good for our health. Uh, we found that by the application of probiotic bacteria, not only increase the yield, but also increase the different antioxidants like anthocyanin, carotenoids, total flavonoids, total phenolics, and overall antioxidant activity. And this paper we published in Scientific Reports, a Nature Group publication. Our student Musaddiqur Rahman Rahi conducted this work, and Julekha Akhtar Mukta also did similar work. That means that work reproduced and very good tool for the production of strawberry, because in case of strawberry, as it is a new crop, farmers use lots of fungicides and other chemicals which is hazardous, because we are eating fruit fresh, so residue of those chemicals are coming to our body through the food web, and food chain. It is problematic. In our lab, we already developed a bank of probiotic bacteria and developed lots of uh, bioassay methods. Some of the probiotic bacteria we sequenced at the whole genome level, and these are now in the pipeline of application in the practical field. Wheat blast, a big disease which appeared for the first time in 2016 as an epidemic disease with yield loss up to 100%. Here is the symptom, and uh, we tested 650 probiotic bacteria, but four isolate display strong antagonistic activity in the laboratory condition. In the laboratory condition, here is the wheat blast fungus, and here is the probiotic bacteria. So in a dual culture plate, you can check whether your probiotic bacteria is antagonistic to the uh, fungus or not. And here you see clearly probiotic bacterial colony here and fungus is here. So something is released from the probiotic bacteria that inhibit the growth. And we checked under microscope. We found that the polar growth are severely impacted, and hyper branching and cell lysis or different morphological alterations taken place compared to the control. In case of control, polar growth of mycelia or hypha is like than normal, and but here serious uh, deformation is taken place. So this is a preliminary screening. After the screening, we checked whether probiotic bacteria produce any antibiotic or not. So we remove the bacteria and the culture filtrate. After autoclaving, we tested on the agar medium, and we found that in presence of uh, uh, culture filtrate provide the you see fungus Magnaporte or IG triticum petotype, its growth is severely restricted compared to the control. That means probiotic bacteria produce heat stable uh, inhibitory substances, that means antibiotic. And in another paper published by Motoshi Chakraborty, you can find in Frontiers in Microbiology as well as PLOS One last year, we found that Bacillus species produce linear lipopeptides, which are inhibitory against the MOT, that is Magna Porte oryzae. And we found that a pure compound, they are very effective in controlling the fungus in the leaf assay. We tested the probiotic bacteria in the laboratory at seedling stage. Here you see very nicely seedling are protected. Here is the control. And we identified the bacteria by using the whole genome sequencing. Here, whole genome sequencing data are available. And it is very interesting that all the probiotic bacteria that inhibited the uh, growth of emotive fungus, and they are bacillus. And bacillus genus is very famous for producing diverse secondary metabolites, lytic enzymes, as well as they have the power to induce systemic resistance. Some of them can fix nitrogen from the atmosphere, as well as they have the power to uh, mobilize or mineralize the organic and inorganic matter, mineral compounds, insoluble nutrients in the soil, and help plant growth. So growth promotion as well as biocontrol both are uh, possible by using the same strain of bacillus. Here are the details of different genes present in the probiotic bacteria that showed the blast biocontrol activity. We found that they produce lots of secondary metabolites, and they have the gene presence in their genome. Uh, and in some cases, they are a little bit different when we compared their genomic sequences. Most interestingly, probiotic bacteria also produced acetoin. Acetoin, because it contains the acetoin bio ALS-SD gene, three of them, and even hydrogen cyanide. Hydrogen cyanide producing gene was not found in BTS3, but other two contains. LSD, this gene, the product is acetoin, and acetoin can induce systemic resistance in the host plant. That means boost immunity of the host plant. They also produce lots of, enzymes. These enzymes are cell wall degrading enzymes of fungus. As a result, they use multiple uh, multiple mechanisms to suppress the pathogen, that is wheat blast fungus in the host plant. Here you see they also produce antioxidant enzymes, because when fungus infect the plant, superoxides are generated. In that case, these antioxidants might help plants to cope up with the stress. Here is the summary of the mechanism of biocontrol of wheat blast by the bacilli. Here you can see different kinds of antibiotics. They are produced by different strains of probiotic bacteria. It is clear that chemical arsenal, that means diverse group of antibiotics, are produced by wheat blast biocontrol bacilli isolated from the wheat and rice seeds. We isolated them as endophyte of wheat seed and rice seed. That means seed endophytes, they are very powerful in biocontrol, and they are because they can colonize the plant very nicely. So mechanism in summary, antibiotics and lytic enzymes non-chemical mean induced systemic resistance in the host plant. So this is really exciting story and exciting information. We tested them in the practical field of wheat blast, wheat in Meherpur, the hotspot of wheat blast. And we found that wheat blast more than 80% is protected by the probiotic bacteria. And this year, this experiment is going on in the Meherpur. I hope you can see the real picture because we are spraying the probiotic bacteria on the wheat at the flowering stage, because flowering stage, heading stage is problematic for wheat blast disease, because wheat blast is basically, head blast, spike are affected. In the vegetative stage, it is not, the symptom is not so clear and no problematic for the plant. Uh, Please visit the Meherpur to see a real effect of probiotic on the protection of wheat from the wheat blast fungus, because after spraying probiotic bacteria, Sanjoy and his team will obviously artificially inoculate the plant to see how much protection can be possible by the probiotic bacteria. So this is a simple lecture for today, and I hope you can get thorough from the literature, because we have some published literature on biocontrol of wheat blast. And recently, Motushi received an acceptance letter of a comprehensive review on biorational management of cereal blast by using probiotic bacteria and their metabolites, and it has been accepted in the critical reviews uh, in biotechnology, which is a very high ranked journal, impact factor more than 8. Uh, I hope I can share it even before it published in the journal. It has just been accepted. Then you can get lots of structural diversities of secondary metabolites that use as an arsenal, chemical arsenal, to suppress the blast fungus in the some of them tested in the practical field, some of them in the greenhouse. There are limited sources of fungicide against the blast fungus, and blast fungus can mutate very fast. This is why fungicide application often result unreliable, performances. Uh, we need living probiotic bacteria, because if probiotic bacteria produce diverse secondary metabolites and induce systemic resistance, as well as make biofilm on the plant surface to compete with the fungus, and then a fungus cannot mutate all the genes to stop the inhibitory or antagonistic effect of the living bacteria. This is why I suggest, if we can find a very good bacterium isolate, or if we can make a consortium of several antagonistic bacteria, those who are not antagonistic to each other, then the consortium would be the best solution, and it would cover, high diverse mechanistic, effort or mechanistic protection against the blast fungus. So, and it is impossible for the blast fungus to, cope up with or adapted to or mutated for, showing the resistance against the uh, consortium of bacteria. This is why this is an interesting area. So, seed endophyte if we can use two, three, or four as a cocktail, and as they are endophyte of wheat, they should not be detrimental to the environment, and they could protect not only blast fungus but also improve the fitness of the wheat plant against other fungus, other stresses, and improve the growth also. So this is our ultimate goal, and if we can achieve those that goal, we can go for commercialization through an industry partnership. And this is the smart strategy for managing any fungal or other phytopathogens in a biorational way without affecting the environment, because uh, chemical pesticides are always hazardous, not only for the target pathogen but also hazardous for the environment, even for the human health, because some of them are coming to the food chain or food wave to our body. The residual effect is really problematic, and they are also expensive. So alternative thinking of using probiotic bacteria would be a sustainable approach, and it is still, in its infancy, because commercial application of probiotic bacteria as an industrial product has not been established in Bangladesh. So we have to go a long way. Thank you very much, Shafi. Now you can ask a few questions.
Yes, sir. The first thing is so we can also termed as the organic agriculture when we will use the probiotics.
It's true. It's true because in probiotic bacteria, as they are a natural organism and they exist in the nature, they even exist inside the plant seeds or plant tissue. This is why application of probiotic bacteria for the promotion of growth, that means plant nutrition and plant protection, if we efficiently could use them as an alternative to the synthetic chemicals, that would promote the organic agriculture, which is now a big craze in Europe and other countries, because they do not like synthetic chemical residue in their food, because some of the synthetic residue in the food are carcinogenic. Uh, you are right, organic agriculture could be promoted. But we need lots of research to understand the molecular crosstalks between and among the probiotic bacteria and the host plant, uh, as well as the enemies of the host plant. That means phytopathogens. Some of the probiotic bacteria have multiple mechanisms not to ward off or inhibit a single pathogen. uh, Multiple pathogen can be uh, suppressed, and host can be protected. This is why we need lots of study to test. When you have a candidate probiotic bacteria as biocontrol, you need to test whether it has any growth promoting effect, that means additive effect, or it has any effect on other pathogens, or even the insect pest, because some of the probiotic bacteria, I told you, bacillus, induce systemic resistance by the production of acetyl. And once induced systemic resistance is developed, it is not specific. It is a broad spectrum, immune system can inhibit the infection by the other pathogens, as well as even the insect. There are lots of literatures are available. Several lines of evidence suggested that multiple, enemies of the plant are uh, suppressed by the single strain of probiotic bacteria. Any more question
No, sir.
or any comment? Because you are now writing a review on seed endophytes. Are you facing any problem, or have you found any interesting data that you want to share here?
Sometimes I'm facing problems. For example, for industrialization, we don't have too much information, and also. And to arrange a table, then I have to also read. I have to also find. It's a little bit tricky.
Yes, what you need to do, you need to quickly go through the book Basili and Agrobiotechnology. I guess you have the two volumes, volume one and volume two. In volume one, you can find an article from some Boris, very famous uh, researcher on probiotic bacteria or plant growth promoting bacteria, and he listed the all commercial products available in the market based on the probiotic bacteria, because in US and worldwide there are more than $4 billion business using the probiotic bacteria. For example, serenade is a very powerful biocontrol agent. So you can find lots of commercial products available in the market or Western market like US or even in India. But as you mentioned, you need to go through the uh, some book chapters that would help you a lot. This is why best idea to scan the two volumes of my book. Even I can share with you the latest volume that I submitted for the publication by the Springer. So three volumes of the book Bacilli and Agrobiotechnology, you can get huge information about Bacilli. But Bacilli are not only the probiotic bacteria or seed endophyte. There are some more others. I guess you can find lots of literatures. What is important, you need to pick up the right literature. And in case of field application, most of the research are carried out in the greenhouse, and limited researches are carried out in the field condition. Lots of literatures are available in vitro activity, because we know the part of the story, not the whole story, how probiotic microorganism protect the host plant from their enemies. Uh, it is, I told you, the infancy uh, uh, stage if you compare the other discipline, because the complex interaction between the probiotic microorganisms with their host, as well as the other microorganism that is pathogenic pathogen of the host are really very complex, and our understanding is really at early stage and not fully understood. The mechanisms are partially being known. This is why inconsistency in the practical application is often reported, depending on the environment, depending on the soil, or depending on the crop. The efficiency of a commercial product of based on probiotic microorganisms are hugely varied. We need to understand the ecological relationship, that is evolutionary co-evolutionary relationship between the probiotic microorganisms which we are using, whether compatible to the plant where we are targeting. Uh, and this is very important. Uh, in this reason, in my team, we are focusing probiotic seed endophytes or endophytic bacteria to develop a biocontrol agent or a biostimulant or a plant growth promoting, you know, uh, agent, because endophytes are, you know, evolved in a way uh, uh, they yeah, are compatible to the host, and they have the crosstalk and, you know, mutualism and symbiotic relationship with the host. So if you artificially modulate their population by using root dipping in the bacterial suspension or seed priming, they easily sustain because they are already adapted to the host and get the carbon and other energy sources from the host plant and give the alternatively or in response to the interacting with the host, they offer lots of benefit to the host. And this idea would be, I think, marvelous. So what you need to do in our campus, uh, with flowering probably being started and border plant which are not experimental, you can spray some probiotic bacteria in limited number of plants repeatedly, for example, every 3 days, and check those who are not sprayed and those who are sprayed whether a seed contain different population of bacteria, because we have a target to load probiotic bacteria in the seed so that it could be a nice product, heavy loaded beneficial probiotic bacteria containing seed. And in our lab, we can quantify by using metagenomic study, we can quantify the bacteria you applied, whether it is loaded in the seed compared to the metagenome in the control plant. So please do it, and especially use the wheat endophyte already we have identified, and that case study would give us more insight about the commercialization of the probiotic in the practical field, especially in the seed industry. So please do immediately design the experiment, because with flowering probably been started, and every 3 days, for example 10 days, if you spray at the flowering stage, then during developing seed, the probiotic bacteria may colonize internal tissues at a higher rate. And if it happens, that would be a great news for the science, and this hypothesis should be tested by you. Small experiment you can do.
Yes, sir. We will do this thing, and we already extracted DNA from the wheat seeds, and within recently we will extract DNA from the roots, and I'm also writing the protocol for that.
Yeah, you need to extract DNA from the seeds, from the root, from the leaf, and also from the spike, and finally from the seed,
From the
developed
next
seed.
year. Okay.
So we can then follow how probiotic bacteria moves from seed to the plant, different organs, even seed to seed, and that would give you a fantastic, information. In that case, uh, please do not mix up the information of the variety, because same variety we need to compare the microbiome or bacterial population or bacteria in the same species, how they are moving in the same variety. Have you got the point?
Yes, sir. We got the point.
Okay, then be vigilant, because our experiment is moving very fast. They are living, so you need to collect the sample in the right time. Even a spraying experiment for the seed loading of bacteria, you have to visit the field and immediately culture enough bacteria and spray regularly to load the seed with the bacteria. And if it happens, that would be fantastic. We can do classical method as well as genomic method to compare the treated one and untreated one. You can target any plant wheat variety, doesn't matter, or you can take two wheat variety, for example barigom 33 or barigom 26. These two ideal variety, one is highly blast susceptible, another one is resistant. In that case, we can compare the data and it would be a nice simple story for publication. So what you need to do, you need to replicate, for example, five plants or you treated and five plants you keep untreated, like that for each variety. And when you spray, you need to spray heavily so that whole plant is uh, you know uh, uh, how to say become wet enough. And evening is the best time, so that whole night the bacteria would get enough moisture to colonize and replicate, and that would be very a good idea, right?
Yes, sir.
By the way, is it possible for you to communicate with the students of 5 to 7 whether we can organize a lecture this evening? Because this evening I am free, but tomorrow, whole day, even the evening, I will be very busy because I am a member of the core committee of crop biotechnology. We have a very important meeting, and then in the evening, we have a seminar with our agriculture minister as well as Shaikh Shiraz, that is Ecological Society of Bangladesh. So please contact with other students, and if they agree, then this evening I shall give a short lecture.
Okay, sir. So 5 to
7.
7. Okay, sir.
BTL 5 to 7, and we can have a lecture 7:30 or like that for 45 minutes, 30 to 45 minutes.
Okay, sir.
Thank you so much. Wish you good luck.
7:30.
Yes,
Okay, sir. Thank you, sir.
all the best wishes.